Welcome back to Quantitative Neuroscience and Mathematical Method in Neuroscience, and this is week 6. I'm giving you a very brief overview of this talk because I want you to have more time on playing with the model than just listening to me. But then I thought you might need some kind of guide, so this is like a brief guide on what we have in this lecture. So we are continuing our discussion, and this is part 3. And this is about gene expression, the final destination. Well, actually, this is not the final destination. The final destination will be the secretion of hormones or neurotransmitters. This is what we have done so far. The inflammatory material, LPS, can bind to TLS4, and when it binds, it catalyzes and accelerates the dissociation of the I-kappa B and NF-kappa B complex. As you know, I-kappa B is just a molecule that holds on to NF-kappa B, so NF-kappa B doesn't get activated. So then it becomes dissociated, then I-kappa B is no longer important and no longer necessary, so it gets degraded. In the meanwhile, NF-kappa B will have a chance to get into the nucleus. So we have made this equation, and then we can do some computational modeling using some rough parameter values, and I actually uploaded it in this file in our class website, and you can run through it using Mathematica, or you can convert it so that you can use it for different software. And basically what you have is, if you have LPS coming in, it will activate TLR. You get see a sharp rise, and then there's a slow degradation. It's going to trigger the dissociation of the IK and NK cytosolic component. And because IK in the previous equation had a degradation term, because we assume there's a degradation, because of that it goes down, right? So it doesn't last forever. One shot of LPS doesn't activate, doesn't dissociate this forever, right? So it goes down, but in this model, the problem is the cytosolic NF-kappa B will go up and sustain. So it permanently turn on, right? And because a portion of it will get into the nucleus, and that approximately half of it will go into the nucleus in our formulation, and it continues on. So they never turn off. So there's something wrong with the equation. What do we do? When there's a dissociation, and of course IK kappa beta get degraded, but portion of NF-kappa B will be translocated into the nucleus. So this much will be lost, right? And that much that being lost must be described in here. The second thing that we need to describe is that they can once again form a dimer and come back to the complex state, right? And then by the amount of this dimerization, you will lose the NF-kappa B free NF-kappa B. So we have to include two terms. First, to take into account the translocation, and the other, the dimerization. So we do that. But we make an assumption that coming back to the complex is a spontaneous process, so it can go back and forth for free. There may be some enzyme catalysis for this, but we do not really suppose it at this moment. Then if you consider that, of course you can write the same thing for the complex formation, but we're not really writing down any complex formation for equation, because we are supposing that there will be a lot of such complex already. So what ends up having is that for the amount that is used for the complex formation, will lose, right? And that much should be added to the both I kappa beta and also NF kappa beta. So it comes down to be like that. So we write down the terms that are lost by dimerization, right? We do not have to write down about this complex itself, but up to this point, we do not talk about the lost NF kappa B due to translocation. But then, still trying to tell you that we make a very important assumption that we have so many of those complexes already, so you don't have to really specify this amount. We are basically saying that there's an inexhaustible pool of the I kappa B and NF kappa B complex, so you don't have to. You have to think about the lost amount in the free NF kappa B and I kappa B, but you don't have to really think about how much are being made into a complex. Then yes, finally, yes, I did think about this. We think about the amount that is lost by nuclear translocation. If you assume that this is a passive process, then of course it just freely goes into the nucleus or comes back to the cytosol, and you just write down the amount that is lost by diffusion, right? And you do the simulation, adding these two new terms, this one and this one, right? Then the result is simply that it can turn off. But the problem is, even if they turn off, those three are exactly the same kinetics. That's really because we have a passive, a loose. There's no time gating for the cytosolic NF B that kind of filters and give a extra temporal kinetics when it enters the nucleus. Biologically, the whole purpose of doing it is to elongate the duration of this protein in the nucleus such that it has a higher chance of getting transcribed, right? One way to do it is to use the switching property of this Hill equation. So what actually happens is this. Translocation is not a passive process. It's not diffusion. It is actually gated by the phosphorylation. So phosphorylation will take time, and it is, once again, the enzyme kinetics. So you can use the same formulation, use the same Michaelis-Menten kinetics. And if you do that, you can indeed make the duration within the nucleus longer. So this is biologically relevant. Now, there's a philosophical point to make. Do we actually really have to use Michaelis-Menten, or can we just make a simplified model that can emulate the same thing? It's your choice. Maybe it'll come down to the point where you have to use a simplified model. Okay. Then you come back, if you finally reach the point where we have to talk about gene expression, what happens is it's the same thing. I'm repeating the same thing. You use the Michaelis-Menten equation again because the mRNA transcription is more or less a process of enzyme kinetics. And then just to briefly go through, it is irreversible, so we use the same kinetics. You can just say that the amount of NF kappa B will lead to transcription of the COX-2 mRNA. And I can also say that for the protein translation, it can be an enzyme mediated process as well, but it is not strictly necessary, right? Because we have so many ribosomes. So the little bit of COX-2, which is a substrate, if you have a little bit of substrate compared to the amount of ribosome, the enzyme, you are dealing with the kinetics at the foothill, right? And on this foothill is almost like being linear. So you don't have to use this complicated mathematics. You can just linearize it. and that's the whole point. And you can just put together. So COX-2 is eventually created, and once again, COX-2 is sort of like enzyme interaction once again, and AA will become a PG, the prostaglandin, through the catalysis of by the COX-2, and it can be exactly written in the microsomatic form. So this is what you have so far, and then finally you can come up to this complicated equation. Now, we didn't show the simulation result up to here. We only showed up to here. So it will be up to you to continue and get the final result, which is prostaglandin concentration given the LPS stimulus. Finally, what I wanted to add is that instead of using these chemical equations, when you're doing computer simulations, you actually have to write down the variables, and that variables can be named in a certain scheme. And I'm giving you an example. And then this is the basic scheme that I would like to introduce. And when you do mathematical simulation, you just my suggestion is to start with a ballpark parameter. So you have to start from somewhere, but then you have to fine tune such that given the input, you get the right output, and you are probably in the right range, right? But then everything, your parameters can be scaled to the biological to make a biological sense. So time can be in the order of millisecond, and we already said that for LPS, the total receptor 4 binding affinity is about a nanomolar, and we have those information all over, how much are there, and but for the mRNA, it's a bit difficult to actually measure. So it's difficult to find the amount of mRNA created de novo for this particular system, but we can use a general idea that is measured for a different system. The basic idea we have is that a single cell contains 1 picoliter of water, not picomolar. So a de novo mRNA usually makes about 1,000 to 10,000 copies of RNA, and that amounts to, if you just divide this multiplied by Avogadro's number, and divide by the picomolar or picoliter of water, then it amounts to about 1 to 10 nanomolar of mRNA. It's quite a lot. And then this amount will become a protein. But what happens is with one mRNA copy, you will have 1,000 protein produced usually. This is just a biophysics. So we can guess that it will be, at the end of the day, 1 to 10 micromolar of protein. And that protein will catalyze and produce prostaglandin. That's a major idea. Finally, I'd have to tell you that there is a term paper. And we have only one term paper required per semester. I'm going to give you 3 to 4 topics. So you don't have to do this one. There are other things to come. So if you like this one, you can start working on this. So basically, you have to start with a ballpark estimate, and then you have to finish the model. That's already a 100 point. But if you want to get extra, think about different sets of parameters that give you the same result, and choose which one makes the realistic parameter set. And that's already enough, actually good enough for a paper. You can actually go for a paper, go beyond the term paper. And finally, think about if the equations are too much sufficient or too less. If this is too less, add more equations and justify it, and things like that. That will be due at the end of the semester. It's completely your freedom to choose. But this is where we are now. We just finished this one. And next week, we'll review the whole thing, do the discussion on the My2TMU board. For this one, we will not have an actual class. We'll just do the discussion online. Thank you very much.